Hello, everybody, again. This is the second talk of our day today. Essentially, what we're going to do is go from my whirlwind overview of the niche modeling process to a detailed worked example. The example we're going to give you is monkeypox in the Congo Basin. So my former student and longtime friend and colleague, Nakazawa, has agreed to give you this presentation, and Yoshi is tuning in from Mexico City. So, Yoshi, I'm turning this over to you, and I will be here just quietly.
Hello, am I on?
You're on. Go for it.
Okay. Thank you very much, Town. Hello, everybody, and I'll be giving you a little example of how we use ecological niche modeling in the public health setting, especially in monkeypox. I'll start with a little overview of monkeypox because I assume not many of you are very familiar with this virus, but then after that I move on to the disease part and the niche modeling part. Let me share this. All right, there we go. So for those of you that are not familiar with this, monkeypox is an orthopox virus that is within the Poxviridae family, which is basically divided into subfamilies: Cortopoxviridae, which can be found in vertebrates, and Entomopoxviridae, which can be found only in insects. Within Cortopoxviridae, there are 4 genera that have members that have viruses that have been found to be able to infect humans and cause human disease. And these are orthopox virus, the ones that we're going to be focused on, parapox viruses, yarapox viruses, and monkeypox viruses. Within orthopox viruses, there are several species that have been described. The most famous and most important one in terms of disease to humans is variola virus, which is a causative agent of smallpox, and it's been eradicated from humans since the late 1970s and 1980s. After that, the one that is currently probably arguably the biggest public health concern based mainly in Central and Western Africa is monkeypox, the one that is the focus of our talk. And there's several other species in causing human disease in Eurasia, which is cowpox, and South America, vaccinia. In North America, there are 3 species that have been recognized and described, but they have not shown to cause human disease, and these are raccoonpox, skunk-pox, and pox. So monkeypox is a zoonotic disease caused by monkeypox virus, which is a brick-shaped virus, which is pictured here on the right picture, and it's a double-stranded DNA virus, which has about 200,000 base pairs in its genome, and of course it's very very closely related to vaccinia, variola, cowpox, and other orthopox viruses. And one aspect that is very important within this genus is that members within this genus they have cross protection. So if you're infected with one of those viruses, you have some certain level of protection to the other viruses. And this is very important because this is what was exploited during the smallpox eradication campaign when vaccinia virus was used as the vaccine. So coming back to monkeypox, the first time it was isolated was in 1959 from a colony of monkeys in Denmark. But the first time that was recognized to cause disease in humans was in 1970, so it's 11 years after. It looks very similar to smallpox in terms of the course of the disease, how it presents in humans. You can see the pictures of this little kid with all the poxes, all the rash, the fever rash in its back. And the only, well, one of the differences is that it's a more benign disease. The mortality rate is not as high as smallpox, but it's still a public health concern. It probably goes to 10 or 15% mortality rate. Although this disease has been recognized for these many years, there's not much that is known about its reservoir in wildlife or its host, and how is this virus maintained in nature. But it is known now that there's two clades of monkeypox virus. One that is basically in West Africa, in all the countries like Liberia, Ivory Coast, Ghana, and Nigeria. And then there's the other clade that comprises all the viruses that have been isolated from Cameroon, Gabon, and mainly in the Democratic Republic of Congo, DRC, where this disease is causing disease in humans. Well, it's very it's endemic in that area, and it's been recorded and seen many times, most of the time until present. There's differences in terms of the presentation of the disease. Also, it's not only genetic. The strain or the clade in West Africa is more benign. It does not cause much mortality, not as high as 10 or 15%, and there have not been many documented transmission chains that go farther than maybe 2 or 3 people, maybe even less. The highest one recorded for Congo Basin is, I think, is up to 7 transmission events from human to human, and that's the highest ever seen, and it's not very common that it happens. You've seen these slides in previous presentations, in Town's presentations. Have you looked at different talks from the biodiversity informatics curriculum? You've seen this. It is the pound diagram, which not going into the details, but it ties the biotic, abiotic, and movement aspects of species. And in this case, when we look at monkeypox, we have a picture similar to this, in which all these points in the map come from or represent the realized niche, which is the combination or the intersection of these 3 major factors: biotic mobility, biotic interactions of the virus with its components. And I put these pictures and maps on the right just to make the point that there was an event of introduction of monkeypox virus into the US in 2003, and this is a clear example of how a virus can be transported or introduced into an area which normally is not accessible to them to it, and human has made it accessible by importing animals. In this case, were animals that came from Ghana, and some of those animals were infected with the virus, and they were co-housed with prairie dogs. So that's a new biotic interaction that it's evidently that the virus can infect prairie dogs because of this event. They were infected by the other animals, from the other animals with monkeypox, and then these prairie dogs were sold as pets, and their owners, the people that got them as pets, started showing up in hospitals with monkeypox in them. It was the western clade of monkeypox because the animals came from Ghana, and it didn't become a huge problem, but it's a clear example of how human can impact this, the interaction of this, can aid the virus to find other hosts that are permissible, but they are not reachable by them because monkeypox virus is in Africa, not in the U.S. This is kind of a very cartoonish illustration of how the monkeypox cycle will be in the Congo Basin. There's hypothetical reservoir or reservoirs in the lower part. The virus will circulate there and then jump into humans or monkeys or other animals that are permissible for the virus. And what we can only see so far, or we've seen until now, is only the human side of it. All the cases that happen that happened in humans, and this is one of the questions that was answered yesterday by Town, talking about how to simplify these very complex cycles. And in this case, it's not that we want to simplify, but it's all the data we have. We only have the data from humans, and we can even simplify this and just take human primary cases that jump from that represent the jump from wildlife into humans, and not the human-to-human transmission, because this human-to-human transmission will obey different rules that may not be caught by ecological niche modeling. This will obey more social aspects of how humans interact with each other, where do they have contact with each other, and how densely populated are places. So we'll definitely, for this talk and from this side of studies, we'll focus more on human to humans, from wildlife to human infections. This is the first, probably the first exercise that was done with monkeypox and ecological niche modeling. Was Town and some other colleagues, Ryan Lash and Christine Ellis, compiled this database which represents all the cases that were recorded by WHO at the end of the smallpox vaccination campaign. So these people, and sometimes these people were presenting in clinics, and sometimes people were encouraged to present to clinics to get diagnosed, and those records, those cases were recorded in a database, and they have some geographic data and the results, if they were basically monkeypox or not. So they they use this database of monkeypox cases with some environmental variables. So in this case, were climatic variables, some soil aspects, and remote sensing data, and came up with this picture, with this potential distribution of this potential distribution of monkeypox in the whole continent in sub-Saharan Africa. But then this was well, this is a very good picture of how where do we could we expect that monkeypox can be transmitted into humans. Later, there have been other other works and other researchers, other groups of researchers that have followed the progress of monkeypox during the last years, during the last 30 or 40 years, and they have shown that there's a possibility that monkeypox cases have been, the number of cases have been increasing during this time, and they have attributed that to the fact that smallpox vaccination is not going anymore. It was suspended at the beginning of the 1980s. So that, because of that, a lot of people have been, well, that the people that have been born after that, they were not vaccinated, and now they represent a population of people that are susceptible to be infected by monkeypox, and that's the reasoning behind, well, their reasoning behind the increase of monkeypox cases. From our point of view, we thought that there could also be an environmental aspect to this, that some places were in 1970s and 1980s where monkeypox was not present. The environmental aspects there, environmental characteristics there, were not suitable for monkeypox to transmit into humans, and maybe currently they are suitable. So we took on this question and used the same use this database, which is from 1980s, 82 to 86 probably, that has all the reports of monkeypox. And Ryan Lash, one of our colleagues at CDC, looked into this database and looked case by case and identified the exact locations where these people had lived and how were they what were the if they were identified as primary contacts, like human wildlife to human infections, or it was a secondary infection like human to human infection. Tom has talked about how important it is to find to curate the data and make it as precise as possible geographically, and that's was a very important part of it. That he has also talked about, or there he talks about testing data, testing the models, and training and testing the model. And the way we did it here was to separate our data into two databases, two subsets of data points. One is represented with the circle dots, and the other one is represented by the blue squares. So we could train the model with one of the databases, and the other one will be our testing databases, and do the reciprocal analysis to test our data and evaluate it. He also talked about environmental data. In this case, we decided to use NDVI, which is a vegetation index that represents greenness of vegetation, or in other terms, how healthy is the vegetation, and um, it also gives us an idea of how productive is the vegetation or how humid it is in that particular time. And so we downloaded, we obtained data, biweekly data for the days, for the years between 1982 and 1986, and summarized data into composites that represent monthly means, maximum, minimum, and range values for this period of time. And we did the same with the data, with the NDVI data for 2002 to 2003 to have a similar database in more present time, in a present time, and that we can use to extrapolate our models. But let's focus first on this first example. So we run the models, and you see that both on diagonal and off diagonal, both databases created models that predict. The one in the left is made by the circle. The model was trained with the circle dots, and it was evaluated with the square points, with the blue squares. You can see that, and the other one is the reciprocal. So it was trained by with the blue squares and evaluated with the white circles. So you can see that both made a good job predicting the other one, which is basically an independent database that is not used in the training. And in this case, we use the modifier ROC, which Tom just talked about, and it's probably going to be part of other talks later. So I'm not going to get into details of that. But once we were sure that we're able to capture a signature, an environmental signature with our data and our niche algorithm, we decided to extrapolate. So on the left, we have our model made in the 1980s with the data and environment, with the case data and environmental data from that period of time that are matching, and extrapolated that into more recent data, 2002 and 2003. And those stars in the right map, in the right figure, are cases that were recorded during that time. Some of them were up to 2009. The arrows, the blue arrows, represent some other cases that were reported, monkeypox cases that were reported after I made this figure. That's why I put these arrows in it. But they all fell in the predicted area for suitable environments for transmission of monkeypox. So if we compare these two maps, sorry about this picture, it didn't translate very well. The stars are still the cases that were recorded in early 2000. The gray area, or it looks kind of white, is the areas where there was no change in suitability, that they were suitable for transmission of monkeypox in the 1980s, and they are suitable in transmission of monkeypox in the 2000s. The red area represent the areas that were not suitable back in the 1980s, but they are suitable now. The blue areas are the opposite. They were suitable in 2000 in 1980s, but they are not suitable anymore in the 2000s. So there, if you look very closely, and I know it's hard to see, but there are some stars that fall in areas that are marked as red. So they were not suitable back in the 1980s, and they are suitable now. If we look at their environmental conditions in those places in these two periods of time, 1980s are represented by the diamonds, gray diamonds, and the 2000s are represented by the black crosses. You can see that the main differences that I will see is that the maximum value of NDVI is maybe a little lower, but the minimum value of NDVI present at those localities are higher now, and the range of values at that particular place through the year are very, very low. So that kind of give us an idea that monkeypox needs, or the reservoir of monkeypox needs environments that are fairly constant because of the low range values, but that they should not go below some value of NDVI. So that is giving us a little bit of idea of what would be affecting either the transmission of monkeypox into humans, or the hosts or reservoirs that carry the virus in the Congo Basin. So here we have seen that we can, or our algorithms can pick a signature, environmental signature, that can be used to find suitable areas for monkeypox transmission. We saw that there might be some expansion of the distribution of these suitable areas into northern DRC, or Southern Central African Republic, and that will also could be contributing to more cases of monkeypox that are being reported now. Besides having that increase in susceptible population of humans that come from the lacking of vaccination against smallpox. So I wanted to talk a little bit about another example of how we have used monkeypox in other situations. This one is an outbreak of monkeypox that happened in 2005 in Sudan, and when this outbreak happened was very surprising, because it is the only outbreak of monkeypox that have been reported in Sudan. There has not been another outbreak reported before, and has not been another one afterwards. And we know that Sudan is probably environmentally is not very similar to DRC, where we know, since we know that monkeypox is related to the rainforest in Central and West Africa. So we looked into it, and these are some of the details of the outbreak. So it happened in October 2005, and it was an 8-month-old child that was involved in this outbreak, and was the first case recognized in this outbreak. This is just to show you where we're talking about. So Sudan is here, or what it was Sudan in that time is here, and Nuria is the place where this case was recognized, and some they went to the hospital, or he was taken to the hospital by his mom to Bentiu. And so it's this whole area where this outbreak happened. And a retrospective investigation showed that there were several suspected cases. Not many of them were confirmed. The two cases that we're talking about are right here, is the little baby and mom, and there was another confirmed case that was probably a horizontal transmission too. The index case was not found, but it was data had been collected and pointed out that this person was sick, and he went to a traditional healer, tooth extractor, who saw him, and after that he went on and cared for other people. So he was probably the person that infected these other people, the point at the source of infection for the outbreak. So another point that has been talked about a lot is the how humans can affect distribution and dispersal of pathogens. So it is important in this case. It was very important to look into how was that area in terms how was that area. What was the situation in that area in terms of politics, in terms of human movement? And it's very interesting because at that time Sudan was coming out of its second civil war that lasted from 1983 and 2005, and they have signed at the beginning of the year 2005 a comprehensive peace agreement that gave some sort of independence to Southern Sudan, to Southern Sudan, and they will come back in 2011 and make a referendum and see if people wanted to have autonomy from Sudan and become two countries that we now know that happened, and they are now two countries. So, but since they were in a civil war, you could imagine that there were a lot of people that flew into neighboring countries as refugees and find safer places to live during this conflict. So once that peace agreement was signed, you also can imagine that people would like to go back to their country and come back to the place they were born or they were more familiar with. And so there was an influx into Sudan and southern Sudan from all the refugees in neighboring countries, and also people were moving between southern Sudan and northern Sudan because they want to be closer to their places where their family were or where they were born. So that could be you could imagine that from the DRC, the Democratic Republic of Congo, some of the refugees, mainly from the northern part of the country, and some of those areas where monkeypox could be present or is present, could have gone into Sudan and gone back. So we talked about this. They had the referendum in 2011, and now there's two countries, Southern Sudan and Northern Sudan. This is the area where the monkeypox case outbreak was reported, and represented with the red star. That's Nuria, the village where the monkeypox case was reported. Zooming into this area, there was another this area represented represented with this rectangle is basically the area in the other map, the map that I have on the left. During that time, there was also a lot of agitation caused by the Lord's Resistance Army, that was basically based in northern Uganda and southern Sudan, and they also and coincidentally or not, during October, the beginning of October or September, late September, they caused they were moving from northern Uganda into southern Sudan and getting into northern DRC, into this national park, going through some villages and just causing trouble in those villages. So you could imagine that people in this area will would fly into or go to somewhere else, going north to get away from this army, because it is a very violent army. So that you will just go away. So that will accelerate people to just move farther, and that could probably be one factor that made people go somewhere northern and go very, very fast. So that's how the situation was there in southern Sudan in terms of social stability. We then went back to our ecological niche models. We make some models with what we know about monkeypox, where it is transmitted. It's basically these black dots. You can see there are nowhere near Sudan, which is this star. But so in terms of geography, there it is very, very, very far from where monkeypox, from where we know monkeypox is being transmitted or is being seen. So we looked into our environmental space to see if maybe those conditions in Sudan, although the place is very far from where we see monkeypox, maybe the environmental conditions are not that different. So we plotted our three principal components, the main three principal components in here, based on environmental variables. The black dots represent all those environments that were that are suitable for transmission of monkeypox. Our model was built on the red dots, the red circles. The gray points are all the environmental conditions that are available in our study area, and the green dots are all the way out here, very, very far from our suitable conditions for monkeypox transmission. So this is another evidence that it's probably it is in a very, very distinct area. What we the other source of information that we normally use in studying pathogens or diseases is genetic information. So we know that within this Congo, within the monkeypox viruses, we have two clades. We have the Congo Basin clade and the West African clade, and we know that there's some difference genetically. So but environmentally they are not that different because they all grew up in this area. So if we translate that thinking, don't know if it's very valuable, if we translate that thinking to southern Sudan, we probably expect to have more genetic variation in a virus that lives in a very different environment. So we did that analysis, and we can see that West Africa, closer together, of course, Congo Basin cluster in this side, and Sudan ends up being right here, very, very close to Yandomi, which is up here in the map, marked as 10, number 10, and the next one will be Mindembo, which is marked with number 8, which is really close to number 10 and number. So genetically, this is showing us that the isolate in Sudan is very, very, very similar to what we can find in DRC. So putting all these ideas together, that geographically it is very far, environmentally the outbreak happened in a place where environments are very, very different than where we normally expect monkeypox to happen, and that genetically the isolate from that outbreak is very similar to some isolates from northern DRC, and that there's the conditions that people could have moved very fast from an area with monkeypox, with circulating monkeypox, into southern Sudan. Our most likely scenario for this outbreak was that it was an importation event of the monkeypox virus. That probably that index case, which visited the doctor in the first place, was probably infected somewhere in DRC and then moved very quickly to Nuria and caused that outbreak in southern Sudan. So with that, I think that's all I have, and I can turn it over to Town, maybe?
Yep, I'm here.
Okay.
Thank you, Yoshi. Well, everybody, so that was intended as a detailed worked example for you. So you've seen several of the techniques that I was outlining in the first talk, and now you've seen them put in dots and rasters on maps. So thanks again, Yoshi, and what we're going to do is end this broadcast. Everybody send questions for Yoshi. We're going to do 2 more presentations, and then we will come back with answers to questions. Thanks a lot, Yoshi.
All right, thank you, Town.
Bye-bye.
Bye.